Hello, this is an introduction to microbiology. That's a lecture that's in particular for surgical technology students. This lecture is going to be covered in a few different videos. So I recommend downloading the PowerPoint slides first, and then we're going to do a background and introduction basic to micro. Then we're going to talk a little bit about the different organisms, starting with bacteria, and then other microorganisms. And then finally, we'll come back and talk about some of the most common infectious agents, organisms that we see, in particular surgically. First, we're going to start with a little history of the background of microbiology. And the father of micro, Anthony von Leeuwenhoek, he was the one to invent the first microscope, and this allowed him to look closer at pond water. He saw the microorganisms in it moving around and could tell that there were live microorganisms in that water. During that time, the basic thought was that these organisms came about by spontaneous generation, so that non-living material spontaneously created these microorganisms. And they did this or tried to or proved this, and I say proved, but they tested this by boiling different types of broths that were infected with bacterium in sealed test tubes, and then to see if it would start growing bacterium again. And so the ideal is if it's sealed, then it's not going to grow anything after it's been boiled. And so they would boil these broths, leave it sealed, and then let it set for a few days and see if it would stay clear or if it would change. Well, they were seeing change in this broth, and you go, what's going on? It's sealed. Well, there's another side to the story. We're going to get to that in a moment. But due to that, they had this idea of spontaneous generation. Now, with the boiling of different broths, and they used different things to do this with, they had a lot of different results. And so we'll get to that too. All right. Now, we first got this disproved by Louis Pasteur, and he's the one that first came out with the hypothesis that microorganisms are in the air. And so he did this using a famous experiment, the Swan neck flask experiment. And I'm going to go forward just a minute to the to a diagram of this, and then we'll kind of jump back there for the moment. But what he did was, instead of having the broth in a sealed test tube, he made this special flask with a swan neck to it. So the broth is in here. And then this neck of the flask would trap any dust particles trying to get in. So he would then boil this, sterilizing the broth, and let it set. Nothing would grow until he would then tip this over just enough that the broth could touch down in the area where the dust particles are trapped, and then set it back upright and watch it grow. And and then he would see the growth in the broth. So here's the kind of an overview of this. Well, one of the first things he did was filtering the air through a cotton plug, and then dropping that into a sterile infusion and seeing it was growing. And then he did the swan necked flask experiment, as I just described there. And so those two things, he says, hey, I think this spontaneous generation thing is wrong. I think microorganisms are elsewhere in our environment, that they're in the air. So we still had that problem with the swan, with the different results coming from the different broths. And so another scientist in the time came along and explained the differences in results that you were getting. So that's John Tyndall. And what he found is that different types of infusions, different types of broths needed different boiling times. And so some things, 5 minutes, but other needed more. And one of the ones in particular was a hay broth or infusion. And so if you think about taking hay and boiling it in water, to create a broth, so you could have chicken broth, you can have vegetable broth, and they made one with hay. And the hay was the one that was the most resilient of the ones that he was running into. And so at this time, he concluded that microorganisms could exist in two forms, one that's more easily killed and one that is more difficult to destroy. Now, what we'll find as we get to bacteria and talking about the different types of forms of bacteria is that some bacillus, rod-shaped bacteria, will form endospores. And this is a non-living form, a very resilient form of the organism in which it contains the DNA of the bacterium. And it can live for extended period of times in a dormant stage, so in a non-living stage. So it can be in too dry of conditions or too cold of conditions, too hot of conditions. And then when it's placed in an area where the conditions are right, then it will turn back into a bacterium. So this is what John Tyndall was hypothesizing at this point, was that microorganisms could exist in those two forms. And we now know today that that would have been living bacteria versus bacteria in that endospore state. All right, next we want to talk a little bit about classification of organisms. And this is kind of a busy little diagram here, but the main thing that I want to show you is that over time, as we've learned more and more about microorganisms, these different classifications have changed. And so we're currently under a classification system of 3 domains, which is this one on the end. And so that's 3 big groupings. That's bigger than the kingdoms. And that is kind of subdivided into prokaryotic cells and eukaryotic cells. And so even through time here, when we started really looking, prokaryotic cells are your bacterium. And eukaryotic is going to be all your other organisms, your animals, plants, protista, fungi, will all be in the eukaryotic. And so we'll look at the differences of these two here next a little bit more, but you can see that throughout this point of the different seasons of this that are prokaryotes, they kind of changed the name of how they wanted to classify these. And then we finally just went to a 3 domain here where you've got 2 domains of the prokaryotes and 1 domain of the eukaryotes. So here's your 3 major domains. Eukarya. This is all your eukaryotic organisms, which are ones that have cells with a nucleus that is enclosed. And most of these are organisms that also have organelles within the cell body. So they're much more organized organisms, and they can be single-celled or multi-celled. Then our prokaryotics are bacteria and archaea. Archaebacteria is a subform or another form of bacteria. And so both bacteria and archaea are both prokaryotic bacteria. The bacteria, previously kingdom eubacteria, meaning true bacteria, And these are prokaryotes, microscopic single-celled organisms that has neither a distinct nucleus with a membrane nor other specialized organelles. And so they're a much simpler type of cell. The archaea are also prokaryotes. The thing that separates them, and obviously there's some folks that disagree that they need to be subdivided or separated out from the other bacteria, but the archaea are more of a primitive bacteria. These are bacteria that have been around a long time, not changed a whole lot. These tend to live in very extreme environments, environments such as high methane environments, high saline or salt environments, hot environments, high methane environments such as animal intestines would be an example. Most of the bacteria that we will discuss will be in this eubacteria domain. All right, so this is a diagram that kind of shows these subdivisions, and so we've got a line here. Everything here on the bottom is our prokaryotes. And so this is going to be made up of these different types of bacteria. These are also Monaria, is another word for bacteria. Monarians are more Monaria. So you got your archaea, and then you've got your regular bacteria or eubacteria. And then all of your eukaryotic, these that have that enclosed DNA, and more organized and can be single-celled or multi-celled. And these are going to branch out into your different ones. And so you've got your protista. And so this is going to be like your protozoa and your amoebas. You've got animals here, which, all kinds of variety and different classifications. Fungi, which is going to include our yeast and molds. And then all your plant kingdom. And then we have our algae over here. All right, so we've got our subdivisions then. We've got the eukarya. So we've got plants, animals, fungi, which also includes molds, and yeast and algae, and then our protista. Now, I want to take this a step further and look at the different classifications of animals. So I've been talking about domain and kingdoms for the most part. These are the biggest group. So domains are at the very top. This is the widest, broadest, biggest group of organisms. For instance, with domains, you've got eukarya, is all of your eukaryotes. That is subdivided into those different kingdoms, which we've got right here. Plants, animals, fungi, and protista. And then the kingdoms can be subdivided then in phylum, class, order, family, and then finally genus and species. Now, genus and species, we're going to look at that a little bit closer because a lot of things like our bacteria will be named by genus and species. I say this is like your last name. Oops, sorry, that doesn't look really good, does it? Last name, first name. So for a microorganism, when we get down to, or any organism, when we get down to the genus and species, it's going to be the broader genus would be like your last name, and species would be your first name. So we'll look at a few of those in a moment. and then kind of going through that whole thing again about how these get more selective in naming of an organism. You can see up here kingdom animata, so your animals, and then we come on down to a specific phylum that will take some of these animals out. And then of that phylum, we get a class, which will take even more out, and then an order and a family, a genus, and a species. And that allows us to narrow down to a smaller group of very much similar. So, these all have some similarities in the kingdom level, as they're all animals. But so are we, but we're not a bird. And so, in order to get narrowed down to that point and to be able to get to a specific type of bird, then you'll need to know all the way down to the genus and species. So let's look a little closer then at genus and species, this binomial nomenclature, this naming using the two names. And again, remember the first name is the last name. So the last name is the first name. So we can have different types of Staphylococcus, and we're going to talk about what Staphylococcus means when we get to discussing the shapes of bacteria. Cocci is a spherical shape. Staph means that it's in clusters. Streptococcus is spherical shaped in chains. So a little bit of difference between Staphylococcus and Streptococcus. But here again, we can have different types of Streptococcus. So, these two have the same last name. Both of these are Streptococcus. And so we've got Streptococcus pyogenes, and we've got Streptococcus pneumonia. So they're both Streptococcus. So that's their last name of the family Streptococcus, not family, but the genus Streptococcus. And then their first name, pyogenes and pneumonia. And then Escherichia coli, we a lot of times will shorten some of these. So we just call this E. coli, which is a much more common hard name. A lot easier to say than Escherichia. Anyway, you get what I'm saying. Much easier. Just say E. coli. All right, and some of the different types of microorganisms that we're going to look at. The first one we'll look at will be bacteria, but then we'll look at algae, protozoa, fungi. Of course, now algae is under fungi, but we'll look at that. They are different as far as how they interact with people. And then the rickettsia. Let me try that again. Rickettsia, viruses and prions. Prions are one that's kind of interesting, is that it's not cells made up of, it's made up of protein pieces. So anyway, kind of a cool one. We'll get to in a minute, and well, more than a minute, but anyway. Let's go a little bit more and talk a little bit about some of the different things about microorganisms in general. So we've got microorganisms, both free-living and parasitic. Microorganisms, we've got them on us all the time. So we've got them on our skin, we've got them in our intestine, we've got them in our airways. Many of these are good. Some of them are neither good nor bad. And then some of them, can become bad or are bad to begin with. So much a wide variety here, but some of them we need in our body. We need them there in order for our body to function normally. But others, if they're not the right ones at the right place at the right time, can cause great harm. Or some that normally live on our skin If we get those into a surgical incision and they're not in the environment where we want them, they then cause an infection. So some of the ways that these microbes relate to each other or relate to humans, and so a few different ones here. Mutualism, this means that both benefit on each other, and so both will get and depend on each other. Synergism is both will work together to accomplish a goal that if they were separate, they would not be able to do. So they're both needed in order to survive. Parasitism, this is one in which the organism benefits, but the host is harmed. And then commensalism, and this one will also be subcategorized, but this means that one organism benefits while the other or the second is neither benefited or harmed. And then that comes into two subcategories: neutralism, meaning that they can occupy the same area with no effect on each other, and antagonism, where one or microorganism interferes with the growth of the other. So let's go back just one step and look at that with those two terms in mind. Looking at one organism benefits, the second is neither benefited or benefits or is harmed, and they can do that by either being neutrally sharing the space, one's benefiting but otherwise no harm on the other one. So when I'm saying one benefiting, maybe the warmth of where it's located, the moisture level, but there's no major effect on one another. And antagonism is where they interfere. One interferes with the growth of another. All right, infections then. What happens here? So anytime or your definition of an infection is multiplication, multiplication of a microbe in, wow, my mouse stuck. So kind of cool, I guess. Multiplication of microbes in a host is called an infection. So that's your definition of infection. So anytime you get a growth of microbes. One of the subdivisions of types of infections that we'll talk about, and we'll talk a lot about this more when we get to wound healing in our main textbook. That's going to come along with chapter 11. And nosocomial infections, ones that are acquired within a healthcare facility. So your other infections opposite of nosocomial would be community infections, one that you get in the community at large. Nosocomial meaning that they're hospital acquired or healthcare site acquired. So and those can vary in how you get those. Nosocomial infection doesn't necessarily mean that there was anything done wrong by the hospital or the medical staff. It could be a wound infection that was a result of poor practice of asepsis, but it could also mean that a patient was in the hospital and a staff member came in that was sick, and the patient got that illness, or there was a patient in the hospital and a family member of that patient came to visit, and that family member was sick, and so while they're in the hospital, that patient gets an infection. It could mean that they've got an IV site too long and it gets infected. Urinary catheter several days and it gets infected. It could also mean that a staff member is taking care of one patient in room A, and that patient is infected with something, and the staff member doesn't wash their hands good, goes to room B, and the second patient gets infected. So different ways you can get nosocomial infections, all right? Of the most commonly transmitted or the most common infection, surgical site wound infection, you're going to hear this said so many times, Staph aureus, Staphylococcus aureus. Interesting thing, Staph aureus lives on our skin. It is a normal microorganism on our skin. Now, when we do surgery, though, we typically interrupt the skin. And so in order to make a surgical incision, we got to cut through the skin. Well, we'll do preps with an antiseptic solution. We'll prep the skin to reduce the microbial growth on it, but there's no way to get it totally to zero. And then we're cutting through it. So it is, possible then for the staph to get into that wound. And so that's one of those things where we really want to be very meticulous with our aseptic technique, with our preps. And as you get to chapter 12 and talk about draping, we'll talk about IBAN, which is a sticky iodine coated plastic drape that we'll put over the area where we're going to be making the incision. So we call it an incise drape because we cut through it, incise into it. But we'll use it on some procedures such as open heart or orthopedics where we're putting in a total knee or a total hip, and areas where we've got to be even more cautious about the potential infection. We'll use this. So then when the scalpel cuts through that incise drape that's stuck to the skin, it's cutting through a sterile layer and less likely to come in contact with the bacteria on the skin. But Staph aureus is there. There's always that opportunity for it to get inside the body, inside the wound, and the wound's warm, it's moist, and it's an ideal location to make it expand very quickly. All right, so we're going to end the first video here. So this is kind of all of our introductory material. And then next we're going to start looking at individual organisms, starting with bacteria, as bacteria has a lot more information along with it because we're going to talk about, looking at the shapes of the bacteria, ground staining, how we can color the bacteria, how we do a culture and sensitivity. So there's a lot more to talk about on the bacteria than what we'll cover with the others. So bacteria we're going to discuss on its own, and it'll be coming up in video 2.